Hi everyone, welcome to Dento'ZEN. We are continuing with our topic of morphology and physiology of bacteria. In this topic, we have already discussed classification, different types of microscopes, different staining methods, various shapes of bacteria in our previous videos. You can check those videos. We have also started with bacterial anatomy, which we are going to continue in today's video. And in the last, we'll take up growth and multiplication of bacteria in a separate video. So let's continue with bacterial anatomy, in which we are today going to discuss the atypical forms of bacteria. So before starting, quickly subscribe to Dento'ZEN, tap on the like button and hit the bell icon so that you remain notified about new videos. So in bacterial anatomy, we discussed the structure of a normal bacterial cell, which has an outer layer or cell envelope. It has protoplasm and cell wall appendages, which are present on the surface of the bacterial cell. In today's video, we are going to discuss the atypical forms of bacteria, which can be an important short note for your exams. And in the next video, we'll discuss bacterial spores, which can be another important short note. So let's start with the atypical forms of bacteria. Now, a normal bacterial cell looks like this, but sometimes this bacterial cell may look like this or this or this. So what are these forms? They are not looking like a normal bacterial cell. They are not typical bacterial cells. So they are called atypical forms of bacteria. So these atypical forms of bacteria can be of two types: pleomorphism and involution forms, and second one is L forms. L form is a unique name. We'll get to know why they are called L form later in this video, but for now you can remember that these L forms can be of two types: unstable L form and stable L form. So let's get to know these types one by one. So first one, pleomorphism and involution forms. Pleo, as the word says, pleo means different, morphism referring to appearances. So if bacteria appears like this, of different sizes and shapes, they are called pleomorphic bacteria. So bacteria can exhibit great variation in their shape and the size of individual cells, and they are called pleomorphic bacteria. And examples of such bacteria are Proteus and Haemophilus. Now, what are involution forms? Sometimes bacterial cell may look like this. What is this? It is a swollen cell. Yes. So involution forms are swollen and aberrant forms of bacterial cell. They are formed in aging cultures which are aging, and in high salt concentration. Many of these cells may be non-viable, they are non-living, and bacteria which can form these involution forms, the examples are plaque bacillus and gonococcus. So those are pleomorphic bacteria and involution forms, and these forms are formed when there is some defect in the cell wall synthesis of bacteria. So they are called to be defective cell wall synthesis. Now what are L forms? So Klinberger Nobel, while he was studying Streptobacillus moniliformis, he observed certain swollen cells and other aberrant morphological forms of bacteria, and he named them L forms. But why L? Why L form? Now this is named after the place where these forms were discovered. That is at Lister Institute, London. That is why they are called L forms. Now, when bacteria forms these L forms, it helps the bacteria in persisting infection. So they have a role in the persistence of pyelonephritis infection and other chronic infections. So these bacteria, that is L forms, they have lost their cell wall. So they are called cell wall deficient forms. So cell wall is deficient in this form of bacteria. Now these L forms can be of two types: unstable and stable. Let's get to know what are these two types. So first is unstable L forms. Now we discussed that the bacteria will lose its cell wall, and when can that happen? When bacteria is in the presence of antibiotic like penicillin. So when there are certain factors like penicillin, enzymes, so bacteria may lose its cell wall, and it may be because of the mechanism of resistance by bacteria. So it may be due to a mechanism of resistance by bacteria. So this L form, that is cell wall deficient form, forms when there is penicillin, it is maintained. But if penicillin or that factor is removed, what will happen? They will go back to their original. So we can say that these L forms are not stable, they are unstable. That is, they can go back to original when that factor, that is penicillin or enzyme, is removed. So that is why they are called unstable L forms. Now here comes two important terms which can be important short note or viva question. So what are these? So one is protoplast. Protoplast. What is a protoplast? So gram-positive bacteria, we discussed in the different staining methods that certain bacteria, they take up violet color in the gram staining. They are called gram-positive bacteria. So when they are placed in a hypertonic solution containing enzyme like lysozyme, so lysozyme can break down their cell wall and there is complete removal of their cell wall. So this form is called protoplast. So protoplast is gram-positive bacteria in the presence of lysozyme. They lose their cell wall completely and is called protoplast. So this protoplast just contains the membrane, plasma membrane and the protoplasm. So it is a protoplast. Then what is a spheroplast? Now spheroplast is derived from gram-negative bacteria. Gram-negative are those which take a pink color or red color based on the counter stain in the gram staining. So these gram-negative bacteria, when they are placed in such a solution containing enzyme, their cell wall is also removed, but it is partially removed. So that the structure that is formed is called spheroplast. So that is the difference. Protoplast is from gram-positive bacteria, cell wall is completely removed. Spheroplast is from gram-negative bacteria and cell wall is partially removed. Now regardless of the type, whether it is a protoplast or it is a spheroplast, both will have a shape which is called spherical. So these will be spherical in shape. So those are protoplast and spheroplast and it can be a short note for your exam. So we have discussed unstable L forms. Now what are stable L forms? Now when bacteria have become cell wall deficient, when they have acquired L form and they remain like that, they do not want to go back to their original, they become stable in that shape, that is called stable L form. So stable L form is a permanent feature of that strain. It is retained even in the serial subcultures, that is even if we take subcultures from the culture, this permanent feature will be seen always, L form will be seen. And it is said that these stable L forms actually resemble mycoplasmas in their characteristics. So actually it may be the mycoplasmas which may be representing the stable L forms. So stable L forms resemble mycoplasmas. So we have discussed the different atypical forms of bacteria, that is pleomorphic bacteria and involution forms. Then we discussed the L forms which can be of two types, unstable and stable L forms. Also we discussed protoplast and spheroplast which can be important short note and viva question. So let's check what have you learned. So what is a protoplast? A protoplast is a bacterial cell A with cell wall but no capsule, or B containing cell wall as well as capsule, both are there, or C free of cell wall and capsule, both are not there, or D it is uniquely sensitive to penicillin. So which one out of these options do you think is the correct answer for a protoplast? Question number 2. What are L forms? They resemble plants, mycobacterium, mycoplasma, or none of the above. So what do you think? L forms resemble which out of these? So that is all for this video. If you really enjoyed the video, do tap on the like button and share this video with your friends. Keep watching, keep learning, keep smiling. See you in the next video soon. Till then, take care. Bye-bye.